This is part 4 for the lecture for Chapter 14, Disease and Epidemiology, and this will be the last lecture for Chapter 14. Up until now, we have been talking about disease in general, especially in a population, different ways to classify it, and how it moves from a reservoir and is transmitted through a population. Now I want to focus for a little bit on individuals. First thing I want to talk about with an individual are predisposing factors. So things that make an individual more likely to acquire an infectious disease. The first predisposing factor I want to talk about is gender. Gender can predispose individuals in two different ways. The first way that gender affects a person's ability to acquire an infectious disease is by the physical structure of the body. Females are predisposed to UTIs just because of the distance between the urethra, the distal end of the urethra, and the anus. E. coli tends to just automatically migrate out of the anus and towards the urethra, and because the distance is so short, a female is much more likely to acquire a UTI compared to a male, where the distance is much longer. So just by virtue of how the body is structured, females are predisposed to UTIs compared to the males. Another way that gender can predispose individuals to infectious disease is by virtue of the hormones and how they affect the immune system. Males are predisposed to meningitis compared to females. So in the same situation, a male is much more likely to acquire meningitis compared to a female. And that does not have to do with the structure of the body because the meninges are structured exactly the same. So it must have something to do with the hormones and how they affect the immune system. But these are two ways that gender can predispose individuals to infectious diseases. The second predisposing factor I want to talk about is genetics. So the types of genes that you inherit can predispose you to different diseases. This is usually associated with genetic diseases, and the most famous example of this is sickle cell anemia. So sickle cell anemia is where an individual inherits a gene that makes the red blood cells sickle-shaped, so boomerang-shaped rather than the normal round shape. And this causes all sorts of problems and inflammation in the individuals. And people who are predisposed to sickle cell anemia have ancestors that lived in areas where there are high rates of malaria. Because what they have found is there's something about being heterozygous for the sickle cell trait that protects individuals from infections from malaria. So there is a high rate of sickle cell genes in areas where there are high rates of malaria. The next predisposing factor I want to talk about is climate or weather. So where you live, the type of climate or the time of year with the weather can predispose you to different diseases. In the winter, in our area, we are predisposed to respiratory diseases. And they used to think that's because the kids are going to school and coming in contact with each other more often. But now what we found out is that in the winter, the air is drier. That tends to dry out the mucous membranes, which makes us more likely to acquire respiratory diseases because we don't have that mucus protecting us from those infections. Another way that location, where you live, can predispose you to disease is if you live in an area with a particular vector. So we've talked about the mosquitoes. You live in an area where mosquitoes carry malaria, you will be predisposed to malaria. You live in an area where the mosquitoes carry Zika, you will be predisposed to acquiring the Zika virus. And that is the same with other insects like the tsetse fly, which is found in Africa and carries Trypanosoma brucei. So living in those areas in Africa will predispose you to African sleeping sickness. The last factor that I want to talk about, I basically call immunocompromised. So these are different things, characteristics people have or things they do that reduce the effectiveness of their immune system, predisposing them to acquiring infections. So the first part of this is something you don't have control over, and that's your age. So there are two times in our lives when we will be immunocompromised. One is when we're very young, and the other is when we're very old. Our immune system is not fully functioning until we're about 7 years old. So before the age of 7, an individual is much more likely to acquire an infectious disease. And then, of course, as we age, our immune system does not work as effectively. So the older we are, the more likely we are to acquire an infectious disease. Another factor that affects our immune system is diet. Sometimes we have control over this, sometimes we don't. If you live in a country where there is a famine or there is a war that inhibits food production, you may be malnourished, and that can affect your immune system. Or you can be in a country like the United States where there is plenty of food, but if you don't choose to eat a balanced diet, you can be malnourished, and that can affect your immune system. So age and diet are not necessarily things you have control over. Some things you do have control over are things you choose to do, such as smoking and drinking. And everybody knows that smoking has an effect on your ability to acquire respiratory infections, and drinking, of course, alcohol. Also, drinking in excess can have an effect on your immune system and predispose you to infectious diseases. When a microbe infects a person, that person will acquire a disease, and the disease will go through specific stages. So going through those specific stages, the easiest way to look at this is with a graph. On the x-axis is time, and on the y-axis is the number of microbes. And the different stages of disease describe the signs and symptoms, the severity of the signs and symptoms that you see during the course of the disease. The first stage of disease is called the incubation period. So the incubation period starts at time 0. That's where you encounter the microbe, and during the incubation period you have no signs or symptoms. So no evidence of the disease is present. And during this time, the number of microbes is very low because the microbes are getting used to your body as an environment, adjusting to that environment. After the incubation stage is the prodromal stage. Not all diseases have a prodromal stage or prodromal period, but this is a separate stage of mild signs or symptoms. So HIV, when a person becomes infected with HIV, initially they get exposed to the virus. They go through an incubation stage of about 2 weeks, and after 2 weeks they have a set of symptoms that looks like the flu. And after that, the infection becomes latent. Later on, they will develop the actual AIDS disease. Food poisoning does not have a prodromal period. So you would go straight from incubation to the next stage, which is the period of illness. So the period of illness, this is where the individual experiences the most severe signs and symptoms. So this would be the highest fever, the most pain, the most swelling, whatever it is that the person is experiencing. During the period of illness, this is where you're going to get a change in the number of white blood cells. And this is an important thing that happens during this period. So the change in the number of white blood cells. If the number of white blood cells decreases, this is a bad sign. So what it means is that the microbe is destroying the white blood cells, and the white blood cells will probably not be able to overcome the infection. If instead the number of white blood cells is increasing, then this means your white blood cells are responding to the infection and that they will be able, hopefully, to overcome the infection, and the person would then progress to the period of decline. So period of decline, decline refers to the number of microbes because the number of microbes are declining, probably because your immune system is responding and eliminating the microbes from your body. Also, period of decline is where the signs and symptoms are declining. So this is where the fever broke, so your temperature is going back down. It's where the pain is being reduced. Your appetite is coming back, the swelling's going down. So that's the period of decline. The last period of disease is the period of convalescence, and this is the period where your body is recovering from the damage that was done during the period of illness. So during the period of illness, you had high numbers of microbes and they were causing all sorts of damage to your body, and it's during the period of convalescence where you are repairing that damage. Throughout this whole curve, it is possible for an individual to be infectious during the period of incubation, prodromal period, period of illness, of course, period of decline, and even sometimes during the period of convalescence. So just because a person is not experiencing any signs or symptoms in these two periods does not mean that they can't transmit the microbe. So it is still possible for them to transmit the microbe. A special situation for diseases in a population are the healthcare-associated infections or the nosocomial infections. And this is a special situation that occurs in healthcare facilities. This involves basically three situations. First of all, the person who acquires the nosocomial infection is a patient. So it's a patient in the healthcare facility, and by virtue of the definition patient, this person is immunocompromised. So they're in that healthcare facility because they are not healthy, basically. Second thing is the environment. This is a healthcare facility, hospital, nursing home, and in this area, that's where you have lots of microorganisms, a high percentage of microorganisms, because people with infectious diseases often go to the hospital and they bring the microbes with them. Then the last part of the nosocomial infection is the chain of transmission. So this is where the healthcare worker transmits those microorganisms to a compromised host. So again, the hospital is the place that has a lot of these microbes in it. You bring an immunocompromised person into the hospital, and then you have the healthcare workers that are transmitting those microbes to the compromised host. And this can be done by direct contact, indirect contact, and even droplet transmission. And the best way to prevent nosocomial infections is for the healthcare providers to wash their hands in between patients and disinfect all surfaces. As a review for the majority of the lecture that has been posted on these videos, we have gone over classification of diseases, transmission from reservoir to the portal of entry, and then the portal of exit to the individual, and predisposing factors, and then the disease process. So as a review, you should go through the general types of diseases and just try to figure out the classification, the transmission, and predisposing factors that an individual could have to acquire these diseases. And a lot of these you already know. Respiratory diseases tend to be contagious, which is easily transmissible from one person to another. Reservoirs tend to be people, transmission, droplet transmission, direct contact, indirect contact, possibly air transmission, and portal of entry respiratory system. Predisposing factors, smoking is a major predisposing factor, as are other things that immunocompromise an individual, like age, diet, and also the winter. Talked about the winter. So that's respiratory diseases. Gastrointestinal diseases. These tend to be communicable. It can be argued if they are contagious or not, but they tend to be communicable, and I've talked about the reservoir, transmission, and portals of entry for those two. Sexually transmitted diseases. Sexually transmitted diseases are definitely communicable. You get them from other people because that is the reservoir. You need that direct contact to acquire them, but they are not contagious. So just being in the same room with an individual is not going to give you a sexually transmitted disease. So they are communicable, but definitely not contagious. And vector-borne diseases are non-communicable. So if you're treating a person with malaria, you cannot get malaria from your patient. The only way you can get them is by the mosquito injecting the plasmodium into your body. Predisposing factors, living in an area where those mosquitoes are present. So just go through your list of microbes to memorize and practice going over these. And again, certain general groups of diseases tend to follow the same classifications and the same transmission and predisposing factors. For the last part of the lecture, I just want to go over the 3 different types of epidemiology. First type is descriptive epidemiology, and this is actually the very beginning of the study of epidemiology, and it was started by this man, John Snow, and he was a physician in England, and he was very interested in cholera. And he was alive during the beginning of the germ theory of disease, and he had this crazy idea that cholera is transmitted by water, which was a crazy idea back then. So what he did was, after a cholera outbreak in his neighborhood in London, he went around from house to house after the outbreak, and he surveyed everybody, asked them how many people got sick with cholera, how many people died, and the last question was, where did they get their water? And what he found out is that all the houses that had cases of cholera all got their water from the same pump. So descriptive epidemiology is where you go through and describe what happened. So this is after an outbreak, where you go back, do the research, you find out where the reservoir was, the mode of transmission, and that sort of thing. So for instance, for coronavirus, we are in the middle of the coronavirus. So we are trying to do descriptive epidemiology on it, but as the numbers keep changing, understanding the transmission, we won't be able to do a really good descriptive epidemiological study until after the outbreak is finished. Second type of epidemiological study is analytical epidemiology. And this woman is Florence Nightingale, and she's basically the mother of analytical epidemiology. You should have heard of her as the woman who basically started nursing. She was on the battlefield treating soldiers who had fallen. Well, after the war, she became very interested in analyzing why certain people got sick and others didn't. So she spent her time making all these graphs for different diseases, trying to understand what factors predisposes an individual to getting sick or predisposes them to recovering where others don't. So that's analytical epidemiology, analyzing the characteristics of the person to determine if they will acquire a disease or not. The last type of epidemiological study that I want you to know is experimental epidemiology, and this is where individuals are trying to determine treatments for diseases. So experimental epidemiology, you are testing out treatments to cure diseases or treatments to prevent diseases. So this involves experiments where you use a drug or a procedure, and you also have a placebo in your control group to determine if that drug is effective. So this is experimental epidemiology. And this is the end of the lecture for Chapter 14.